Welcome, comrades perennials, to another episode of the audio podcast Synthetic. Artificial intelligence will not replace you, but someone using artificial intelligence definitely will. So, if I told you that just drinking a few extra cups of coffee every single day could, radically reshape your body,
Right.
like literally shifting weight away from dangerous organ fat, moving it over to skeletal muscle, and doing all of that without you losing a single pound on the scale.
Yeah, it sounds completely made up.
It really does. You'd probably think I was pitching you some terrible late-night infomercial supplement. But a massive, decades-long population study out of Finland just proved exactly that.
It's incredible data.
It is. So today we are unpacking the hidden molecular mechanisms of metabolic health, insulin resistance, and how daily dietary choices literally reprogram the human body.
Exactly.
Because whether you are, holding a cup of coffee right now or you're just thinking about what to eat for dinner tonight, the molecules you are about to swallow are going to interact with your DNA and hormones in ways you likely never realized.
Oh, absolutely. The molecular level is where the real story is.
Right. And we are bridging this fascinating population data with cutting-edge cellular science to see how the bioactive compounds in our food act as literal metabolic software updates. So let's start with this Northern Finland birth cohort 1966 study.
Yeah, let's get into it.
The sheer scale of this data set is just incredible. We're looking at 2,264 participants, and they're all exactly 46 years old, which is brilliant because it completely removes age as a confounding variable.
Right, which is huge in metabolic studies.
Exactly.
Yeah.
And they live in a country with a massive coffee culture. Like the average person there consumes almost 12 kilograms of coffee a year.
That is a staggering amount of coffee.
It really is. And the data revealed something that completely challenges how we normally think about health and weight. The habitual coffee drinkers had the exact same body mass index, or BMI, as the non-drinkers.
Which is wild.
Right. According to the scale, they were identical, yet their actual internal body compositions were entirely different. Judging your metabolic health purely by BMI is basically judging a book entirely by its page count, while totally ignoring the actual story printed inside.
That's a perfect way to put it.
Because the heavy coffee drinkers had significantly less visceral fat and significantly more skeletal muscle mass. So I have to ask, how does a simple calorie-free beverage shift fat storage away from our organs and toward muscle tissue? All while a number on the scale stays exactly the same.
Well, the physiological findings from this cohort point directly to biological mechanisms that act way beyond mere calorie counting.
Okay.
The reduction in visceral fat is really the crucial part here. Because, visceral fat isn't just the soft subcutaneous fat located just under your skin.
Right, the stuff you can pinch.
Exactly. Visceral fat is the dense, highly inflammatory fat that physically wraps around your liver, your pancreas, and your intestines. It acts almost like an invasive organ itself, actively secreting inflammatory chemicals.
Oh, wow.
Yeah, and the coffee drinkers had far less of this dangerous fat and significantly more metabolically active skeletal muscle. But honestly, the most revealing marker was a pronounced drop in circulating branched-chain amino acids, or BCAAs.
Wait, BCAAs? Aren't those the supplements fitness enthusiasts take to build muscle?
They are, yeah. But chronically elevated levels of these amino acids in your bloodstream when you are simply at rest, that's actually a massive red flag.
Really? Why is that?
Because they're a well-documented early biomarker for insulin resistance. High resting BCAAs are a strong predictor of developing type 2 diabetes later in life.
Okay, so the fact that habitual coffee consumption naturally lowered these circulating BCAAs, that implies the body is just utilizing its nutrients much more efficiently.
Exactly. Furthermore, there was a distinct hormonal signature uncovered in the data. Like in men, higher coffee intake correlated with more favorable glucose and insulin profile, higher total testosterone, and increased levels of a protective protein called sex hormone binding globulin, or SHBG.
SHBG, right.
Yeah, and this protein binds to sex hormones and regulates exactly how much of them are actively circulating and interacting with tissues in the blood.
Just to make sure the mechanics of that are clear for anyone listening, having more of this binding globulin basically means the body is much better at managing its hormone reserves.
Precisely.
Rather than just, letting free hormones run wild and cause chaos in the bloodstream. And this protective effect wasn't just limited to the men in the study, was it?
No, it wasn't. In women, the hormonal associations followed a similar, albeit a bit more limited, pattern. They also demonstrated higher levels of the sex hormone-binding globulin and lower measures of free androgens.
Androgens are male sex hormones that women naturally produce in smaller amounts, right?
Right. And lowering excess free androgens in women is generally associated with much better metabolic health and a lower risk of endocrine conditions.
That's fascinating.
Now, obviously, because this is an observational study, we are looking at a strong, undeniable association rather than a direct proven cause and effect relationship.
Right, it's an association.
But it firmly establishes that the complex compounds found in roasted coffee beans are actively interacting with the human endocrine system, altering how the body partitions nutrients, manages hormones, and ultimately decides where to store fat.
Okay, so the coffee drinkers have significantly less of this highly inflammatory visceral fat packed tightly around their internal organs.
Yeah.
But why does that visceral fat actually matter at a microscopic cellular level?
Yeah, that's the key question.
We hear the phrase visceral fat is dangerous constantly, but what is that fat actively doing to our cells to cause insulin resistance? I like to think of it this way. If insulin is a specific key meant to unlock a cell's door so that blood glucose can get inside, ectopic fat, which is fat stored where it just doesn't belong.
Right, misplaced fat.
Yeah, it's essentially like someone jamming a wad of gum right into that cellular keyhole. The key can no longer turn, and the glucose is left stranded outside the cell, floating aimlessly in the bloodstream. So how does that physical gum in the keyhole translate to the actual biology of an insulin-resistant cell?
Well, to understand that biological gum, we really need to look at how the cellular lock is supposed to function under healthy conditions.
Okay, let's break it down.
Normally, when you eat a meal and your blood sugar rises, your pancreas releases insulin. That insulin travels through the blood to a muscle or fat cell and docks into a specific receptor on the surface called the IRTK.
Got it, the docking station.
Exactly. This docking action sends a mechanical signal into the cell, activating a relay team of messenger proteins, specifically IRS1 and IRS2.
IRS1 and IRS2, the relay team.
Right. And this elegant cascade ultimately activates the PI3-ACT pathway, which tells the cell to send GLUT4 glucose transporters up to the surface membrane, grab the sugar out of the blood, and pull it inside to be burned for energy.
Okay, that makes sense. So where does the fat come in and break this?
That's lipotoxicity, the toxic accumulation of fat. When you have excess visceral fat, lipids start spilling over into tissues where they have zero business being, particularly the liver and inside the actual muscle fibers.
Oh, so they physically get inside the muscle.
Yes, and once inside, these misplaced fats break down into highly toxic lipid byproducts called diacylglycerols, or DAGs and ceramides.
DAGs and ceramides.
Right. These toxic byproducts are the gum in the keyhole. They act like biological alarms, alerting the cell that its internal environment is completely polluted. They violently activate inflammatory pathways and severe stress responses inside the cell.
So these misplaced fat molecules are not just passively taking up space in the muscle fiber, they are actively rotting the machinery from the inside.
Precisely. They are turning on the cell's internal fire alarms.
Which brings us to the concept of oxidative stress, right?
Right.
Reading through the materials, it sounds like the cells are essentially rusting from the inside out.
Rusting is an incredibly accurate way to visualize oxidative stress. When these toxic lipid byproducts trigger the cellular alarms, they cause severe dysfunction in the mitochondria.
The power plants of the cell.
Exactly. Imagine an engine flooded with bad fuel. It starts sputtering and kicking out toxic black smoke. In the cell, this smoke takes the form of highly reactive oxygen molecules driven by the upregulation of enzymes like NOx4.
Wow, okay, so they're just throwing out sparks everywhere.
Yeah, these reactive oxygen molecules fly around the cellular environment, damaging proteins and violently ripping electrons from other delicate structures, just like rust eating away at metal gears.
That is terrifying.
Concurrently, a state of chronic low-grade inflammation takes hold, driven by pathways like NF-κB and JNK. Inflammatory proteins flood the cellular environment.
So the cell's just drowning in chaos.
Yeah, and here is exactly how insulin resistance happens physically. These inflammatory proteins and the oxidative rust physically attach themselves to the insulin messenger proteins we talked about, the IRS1 and IRS2.
Oh, they directly attack the relay team.
Exactly. They place a chemical tag, a phosphate group, on the exact wrong spot of the messenger protein. Because the messenger is now chemically altered and deformed, it can no longer pass the insulin signal down the line.
So the insulin docks on the outside perfectly, but the message just never reaches the glucose transporters waiting inside.
You've got it. The cellular door stays bolted shut, and blood sugar rises uncontrollably.
That paints such a vivid mechanical picture. The cell is drowning in toxic fat sludge, the internal engine is rusting out from oxidative stress, and the messenger proteins are physically mangled, so they just can't do their jobs.
It's a complete systemic breakdown.
And this cellular damage directly connects to our genetics. Diet does not just provide raw calories or passive building blocks. It actively changes how our genes are expressed without altering the underlying DNA sequence.
Epigenetics, it's a game changer.
Right. If human DNA is the hardware of a computer, the physical hard drive you were born with, epigenetics is the software operating system running on top of it.
That's a great way to think about it.
The bioactive compounds in our food, or that toxic fat sludge we just talked about, are essentially acting as daily code patches or malware. So how specific do these epigenetic software updates actually get when we're looking at something as complex as insulin resistance?
The specificity at the epigenetic level is astonishing. We are not just talking about broad systemic changes. We are talking about individual genes being manually dialed up or dialed down based entirely on the chemical environment.
Manually dialed. How?
There are three primary mechanisms at play here. First, there is DNA methylation. Imagine the DNA strand has little chemical stop signs attached to it.
Okay, stop signs.
When a gene has many of these stop signs, it is silenced. The body cannot read it. In states of insulin resistance, we see a decrease in methylation on specific genes, meaning the protective stop signs are maliciously removed.
Maliciously removed.
Yeah. For instance, there's a gene called TXNIP, which is responsible for activating a massive inflammatory complex called the NLRP3 inflammasome.
The NLRP3 inflammasome. Sounds aggressive.
It is. It pumps out IL-1 beta, a highly destructive inflammatory cytokine. When the stop signs are removed from this specific TXNIP gene, the body constantly produces high levels of this cytokine. We also see this with SOCS3 hypomethylation, which actively increases type 2 diabetes risk.
So wait, let me make sure I'm wrapping my head around this.
Huh?
The toxic environment created by the diet is literally stripping away the chemical stop signs that normally keep our most dangerous, destructive inflammatory responses in check.
Precisely. It is exactly like an antivirus software being uninstalled, allowing the inflammation malware to run continuously in the background. The environment dictates the code.
That's wild. What's the second mechanism?
The second involves histone modifications. Your DNA strand is incredibly long, so to fit inside a microscopic cell, it wraps tightly around protein spools called histones.
Right, to stay organized.
Yeah. If the DNA is wrapped tightly around the spool, the gene is hidden and turned off. If the spool unwinds, the gene is exposed and turned on. In people with high BMI and severe insulin resistance, we see specific chemical tags, like elevated H3K4 Me3 levels added to these histones in their fat tissue.
So these tags change how the spool works.
Exactly. They physically force the spools to unwind. There's also a process called SET7-dependent H3K4 methylation on the NFKB P65 promoter, which basically permanently exposes genes that drive a systemic whole-body inflammatory state.
The physical structure of how the DNA is packaged is completely altered by the metabolic disease.
It is. And the third mechanism involves non-coding RNAs, specifically microRNAs. These are tiny floating snippets of genetic material that act like rogue editors.
Rogue editors.
Imagine a factory where the DNA sends an instruction manual down to the factory floor. These microRNAs intercept the manual in transit and shred it before the cell can build the required proteins.
Oh, wow. Do you have an example?
Yeah, in visceral fat tissue, a specific microRNA called miR34A is upregulated, and it actively suppresses the body's anti-inflammatory
It
macrophages.
suppresses the good guys.
Exactly. And in skeletal muscle, other specific microRNAs like miR1 and LET7 intercept the blueprints for building those insulin messenger proteins we discussed earlier, IRS1 and IRS2. There's also miR29, which actively shreds the instructions, physically shutting down the muscle's ability to ever respond to insulin.
This means the food we eat and the subsequent internal environment we create is dictating which parts of our genetic code are turned on or off in real time. It's writing microscripts that tell our muscle cells to either listen to insulin or ignore it entirely.
It's all connected.
Shifting focus from the microscopic DNA level up to practical macrodietary patterns, we look at the Mediterranean, plant-based, and low glycemic index diets.
The big three.
Yeah. But the fundamental question for anyone trying to apply this information is why these diets actually work. Are these dietary patterns merely effective because they force people to eat fewer calories, leading to weight loss, and the weight loss itself fixes the cellular machinery.
That's what most people assume.
Right. Or is there an independent structural metabolic mechanism at play that improves health even if the person's weight stays exactly the same on the bathroom scale?
The evidence strongly points to independent structural metabolic mechanisms at play, completely regardless of mere weight loss.
Really? How so?
Take the Mediterranean diet. It fundamentally alters the internal landscape, even in the complete absence of calorie restriction. It actively reduces hepatic steatosis, or liver fat.
Okay, clearing out the liver.
Exactly. By clearing out the toxic fat sludge from the liver, it restores the liver's ability to process glucose and insulin correctly. Furthermore, it completely remodels the gut microbiome. The high intake of specific plant fibers feeds beneficial bacteria, which in turn ferment those fibers into short-chain fatty acids, or SCFAs.
SCFAs, I've heard of those.
Yeah, these fatty acids are powerful signaling molecules that travel through the blood, calm the immune system, and they even directly suppress that NLRP3 inflammasome we mentioned earlier. The diet also interacts with specific genetic backgrounds, essentially providing the software patch to fix the genetic inflammation loop.
It is not just about eating less energy. It is about providing the exact right chemical information to the gut bacteria, which then basically manufacture the custom medicine our cells need.
That's a brilliant way to phrase it.
And I assume plant-based diets operate on a similar principle, just utilizing slightly different metabolic pathways to achieve the same goal.
They do operate through distinct mechanisms, yeah. Plant-based diets fundamentally shift the amino acid composition of the fuel the body receives.
How so?
By their nature, plant proteins contain lower amounts of those branched-chain amino acids, or BCAAs.
Oh.
Yeah.
This ties perfectly back to the findings of the Finnish coffee study.
Exactly. By naturally lowering the dietary intake of BCAAs, you alleviate the metabolic bottleneck that leads to insulin resistance in the muscle tissue.
That makes total sense.
Additionally, plant-based meals elicit a much greater postprandial incretin response.
Incretins.
They're hormones released by the gut after eating that tell the pancreas to prepare for incoming sugar. A diet rich in whole plants significantly boosts the secretion of a crucial incretin called GLP-1, which naturally enhances insulin secretion and slows digestion.
And what about low glycemic index diets?
Low GI diets focus on a completely different vector mitigating postprandial glucose fluctuations. By avoiding massive spikes in blood sugar after a meal, they fundamentally reduce the body's total daily insulin demand.
So you're just taking the pressure off the pancreas.
Right. When the pancreas doesn't have to pump out massive waves of insulin all day, the incretin responses alter and the cells become more sensitive to the baseline amounts. This actively decreases that intramyocellular lipid accumulation, the ectopic fat inside the skeletal muscle cells.
So each of these dietary patterns modifies the internal metabolic environment through highly targeted molecular pathways, not just by creating an energy deficit.
Exactly. It's structural remodeling.
The overarching pattern here is that the overall diet is essentially a delivery system for specific molecular tools. If we look closely at these specific bioactive compounds found within these diets, we are dealing with polyphenols, peptides, and prebiotics.
The microinterventions.
Yeah. We know pharmaceutical companies spend billions of dollars designing highly targeted metabolic medications to fix insulin resistance. Can a simple plant peptide or a polyphenol extracted from an apple actually mimic the biological actions of those targeted drugs?
The biochemical reality is that many of these plant-derived compounds operate on the exact same molecular pathways as targeted medications, and in some cases, they do so with remarkable precision.
That is just incredible to me.
Let's look at polyphenols, which are the chemical compounds that give plants their color and protect them from environmental stress. Resveratrol, which is found in the skin of grapes, quercetin from onions and apples, and catechins, specifically EGCG from green tea, they don't just passively float around. They actively seek out and suppress the oxidative stress we discussed earlier.
They act like cellular mechanics.
Yes, they do. They activate the AMPK and SRT1 pathways inside the cell. When activated, these pathways essentially mimic the physiological effects of fasting and severe exercise, even if the person is sitting perfectly still.
Wait, really? Just by drinking green tea?
Yeah, on a molecular level. They sweep up the oxidative rust and physically restore the broken PI3 ACT signaling cascade, allowing the messenger proteins to function normally again.
Wow. And what about omega-3s?
Omega-3 fatty acids operate on a similar level of precision. EPA and DHA activate specific receptors called GPR120 and FFR4 on macrophages, which halts inflammation and prompts the production of highly specialized molecules called resolvins.
Resolvins, I like that name.
It fits perfectly. Resolvins don't just prevent inflammation, they actively seek it out and resolve it, returning the tissue to a healthy state.
So when you consume an onion or a cup of green tea, those specific polyphenols are traveling directly to your muscle cells, turning on the biological switches that simulate exercise and repairing the deformed insulin locks.
This is exactly what they do.
That is a profound way to look at a standard grocery list. What about the structural proteins in plants? How do they interact with our cellular machinery?
This is where the science gets incredibly compelling. We are looking at plant-derived bioactive peptides. These are specific, short sequences of amino acids locked inside larger plant proteins.
Okay, so they get unlocked during digestion.
Right. When you digest soy, rice bran, hemp, or lupin beans, your stomach acids break the larger proteins down into these specific peptide chains. Researchers have identified precise peptides such as soy-1, the FLV tripeptide, and a glycine derived from soy that have a staggering mechanical function.
Gotcha.
They structurally bind to and inhibit an enzyme in your body called DPPIV.
BPIV, what does that do?
The sole job of the DPPIV enzyme is to break down GLP-1, that beneficial gut hormone we talked about that regulates insulin and appetite. By inhibiting the enzyme, the plant peptides allow GLP-1 to stay active and circulating in your bloodstream for much longer periods.
Wait, inhibiting the DPPIV enzyme to keep GLP-1 active in the blood, isn't that the exact mechanism of action for an entire class of modern prescription diabetes and weight loss drugs?
It is the exact mechanism. Pharmacological DPP-4 inhibitors are a major billion-dollar class of diabetes medications. And these microscopic plant peptides derived from a simple soybean or a beta-congluten from a lupin bean are executing the exact same biological blueprint.
That is mind-blowing.
They also inhibit alpha-glucosidase, an enzyme responsible for digesting complex carbohydrates in the gut, thereby naturally flattening the blood sugar curve after a meal while simultaneously stimulating GLUT4 translocation to bring glucose into the cells.
And you mentioned prebiotics too, right?
Yes. Specific prebiotics found in fiber-rich foods actively alter the gut ecosystem. They serve as primary fuel for highly beneficial bacteria strains like Akkermansia mucinifera.
Akkermansia mucinifera.
Right. When these bacteria thrive, they pump out butyrate, a short-chain fatty acid that travels directly to the mitochondria and forces them to burn fat more efficiently.
It's a completely interconnected system.
It is. And finally, micronutrients serve as the structural scaffolding for this entire process. You have antioxidants like astaxanthin, but also vitamin D, magnesium, and chromium. They are not just generic healthy vitamins, they are non-negotiable chemical cofactors.
They are structurally required.
Yes. The cellular insulin receptor literally cannot physically function without adequate magnesium present to facilitate the chemical docking reaction.
We have taken quite an intellectual journey today. We started with the surprising mystery of heavy coffee drinkers in Finland shifting their body composition away from dangerous organ fat without losing a single pound.
A great starting point.
Right. And we traced that biological thread down into the microscopic world of lipotoxicity, watching ectopic fat sludge jam the cellular keyholes and rust out our mitochondrial engines.
The biology is so elegant, yet so fragile.
It really is. We saw how the exact chemical environment we create inside our bodies writes epigenetic software updates, physically muting or amplifying the genes that control systemic inflammation. And finally, we discovered how the precise, targeted molecules found in apples, green tea, and soybeans can act like molecular mechanics, mirroring the effects of advanced pharmaceuticals to literally repair our broken insulin pathways.
It really reframes how we should look at a meal.
It does. Given that our gut microbiome, our hormone receptors, and our epigenetic markers are constantly adapting to the exact molecules we ingest minute by minute, perhaps it's time to completely abandon the idea of food as mere fuel or passive calories.
I agree completely.
Instead, we should view every meal, every snack, and every cup of coffee as a real-time, high-stakes chemical conversation with our own genome, a daily opportunity where we hold the power to dictate the literal terms of our own metabolic aging. Subscribe to the podcast series Nuclear Locomotive, Neural Sawdust, and Synthetic. Namaste, thank you, and rest in peace. Good luck.